DNA is the coding language of life. From biological processes to essentially all of an organism's characteristics, DNA governs it. For this reason, it's easy to see how important an understanding of DNA would be to medicine, whether it's biological engineering, development of new treatment therapies, or diagnosis of diseases. In the past few decades, through various efforts, we've accumulated a massive amount of genomic data for this very reason, in the hopes that we'd be able to understand it and hopefully use it to our advantage. However, we still currently don't have a fundamental understanding of exactly how the human genome codes for biological function. So that's the goal of this project, to use deep learning to model the language of the genome and help us better understand how it codes for biological function. The central idea behind this project is that the language of DNA really is a language and has many similarities with human language. So I applied the process that humans use to acquire natural language to the language of the human genome. This is done through a combination of convolutional neural networks with a transformer-based language model. So the transformer-based language model is the current state-of-the-art model for natural language processing, so human languages. The current stage of this project, MuteFormer, is focused on one specific facet of genomic function, which is proteins, and more specifically, protein mutations and their effect on disease. The input of MuteFormer is a protein mutation, and the output is its pathogenicity prediction, so the probability that this mutation will cause some sort of disease. Going a little bit into exactly how MuteFormer works. So, as briefly mentioned, the MuteFormer model consists of convolutions and transformer language model body. So the convolutions are used in the adaptive vocabulary of the model. The adaptive vocabulary basically allows the model to identify short-term dependencies, in other words, patterns, within the protein language that serve as its vocabulary. This learned adaptive vocabulary enables it to construct a much better representation and interpretation of every single protein sequence. The language model body then uses self-attention mechanisms to consider context and enables the model to basically identify the syntax and grammar of the language. The training procedure is also similar to how humans would interpret language. So the pre-training phase is when the model actually gets familiarized with the model itself, with the language itself, via being given a lot of sentence examples from that language. The fine-tuning phase is then when the model uses this knowledge to perform a specific task. In this case, the model is fine-tuned for pathogenicity prediction, although in theory it should be applicable to other protein language tasks as well. The fine-tuning results up here. Fine-tuning was tested for 3 different methods on many different hyperparameters. So here we have a comparison of the 3 different fine-tuning methods, the paired sequence being the best one. We also have a comparison of the MuteFormer model with the classical transformer model that was originally used for human language processing. Down here is the pathogenicity prediction performance comparison of MuteFormer versus other models, other existing methods of pathogenicity prediction. So this is a receiver operator characteristic curve. Higher area under the curve is better, so the ones in the upper left corner are the best performing methods. From this graph, we see that MuteFormer matches or outperforms essentially all of the methods here. Pathogenicity prediction by itself has its own applications in medicine, such as identifying potential mutation sites for drug development or genetic screening for diagnosis of diseases. But this, more importantly, also serves as a proof of principle for a general direction of being able to use language models to model the language of the human genome and help us better understand it. So in terms of future plans, I'm currently working on architecture improvements for MuteFormer, including the incorporation of 3D structure. And then in terms of other plans, the ultimate goal for 3D structure is a internally self-folding 3D structure within the transformer model. Also other protein-related tasks, as I briefly mentioned, and finally DNA-related tasks. So yeah, that's my project. Thank you.